Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Scaling Cell Line Development, Enabling IND-Ready GMP-Compatible Workflows. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank Nova Biomedical, who developed the content for this presentation. Advanced Instruments and Nova Biomedical are now doing business under the same brand, bringing together decades of expertise in analytical instrumentation, R&D, and global customer support. The Solentim ecosystem is now part of Nova Biomedical's portfolio, providing best-in-class imaging and single-cell deposition technologies for cell line development workflows and assurance of clonality for regulatory bodies. Now I would like to introduce our speakers for today's event. Adam Kosser is a global product manager for the Solentim portfolio at Nova Biomedical, which includes an ecosystem of instruments that digitize and automate CLD workflows. Prior to this role, he worked in application sciences at both Syneon and Advanced Instruments, specializing in precision dispensing and single-cell isolation for diagnostic test development and CLD, respectively. Sakti Murthy is a lead scientist at CCRM Toronto and a subject matter expert in gene editing and analytical development. She leads cross-functional teams focused on genome-engineered cell lines and advancing allogeneic cell therapies for single use. She has established scalable IND-enabling workflows using multiple gene editing technologies, translating innovative approaches into GMP-aligned, industry-leading processes. But now, without further ado, I'd like to go ahead and hand the presentation over to our first speaker for today. Adam Kosser, you may begin when you're ready.
Thanks, Ryan, for the introduction. So, as mentioned, my name's Adam, and I'm the product manager here at the Solentim portfolio. And I'm just going to give an overview of what we're going to cover in today's webinar. So, first up, we're going to give an introduction to the technology that was used by Sakti at CCRM within her case study, that being the VIPS PRO. We'll then move on to see how Sakti has used the VIPS PRO in practice. And then the last section to wrap up will be about how we can image difficult to visualize cell lines by onboarding off-the-shelf artificial intelligence. So just before we get going, I just wanted to give a high-level overview of what we're referring to when we say the Solentim ecosystem. So the Solentim ecosystem boasts a suite of 3 different instruments: the VIPS PRO, that's going to be the topic today, that's used for single-cell seeding; the CellMetric X, which is used for the high throughput imaging. And then there's also an instrument called the ICON, which is there for characterizing clones and also any proteins that are produced by those clones. All 3 of these instruments are operated through what we call the STUDIUS software. And this is a common data management package which automates the tracking, triaging, and reporting of clones anywhere from the single cell, but also throughout scale up, eventually in the bioreactor at the end. And this is useful for clone selection, but also your clonality reporting, which is 21 CFR Part 11 compliant. Okay, so we're just going to do a bit of a deeper dive now into what is the VIPS PRO and what's the underlying technology that the team at CCRM have onboarded. Firstly, the VIPS PRO, the image that you can see on the left-hand side. So this produces gentle nanodroplets for single-cell cloning. It also has high-resolution optics to image the droplets produced by the instrument, and also whole wells for clonality classification. It has automated liquid handling to deposit the media into the plates, And then all of the consumables associated with this instrument are single use, and there are GMP compliant consumables available as well. In terms of the core technology for cloning single cells, so you would put an empty 384 or 96 well plates onto the VIPS PRO. And what the VIPS PRO does is it deposits a nano droplet of a cell suspension. And that droplet is imaged at 20 different heights, what we would call a Z stack of 20 different images. And artificial intelligence is applied to look at those droplets and determine whether it's empty, contains a single cell, or contains multiple cells. If it's empty, it will retry the droplet and continue up to 16 times or up until there's a single cell that's been deposited. After it's made that decision, it will then automatically fill that well with a predefined volume of media that's set by the user. So this is the end outcome of the process. So these are 3 different images that are produced via the VIPS PRO, and they're not whole well images, they're just image of a droplet in a single frame. And what you can see on the left-hand side is a droplet containing no cells. In the middle, a droplet containing a single cell that's been classified as a clonal cell line produced in that well. And then on the right-hand side, a droplet that contains 2 wells that's been classified as non-clonal. And the benefit of this is as soon as reagent hits the bottom of the well, you have a good idea and a good understanding and high confidence that that well is clonal or not. As I mentioned, it's an artificial intelligence-based neural network that detects whether there's cells present in the droplets. And this network has been developed, trained, and also validated on a variety of different cell lines. A CHO cell line with 96% accuracy that you can see on the left-hand side, but also HEC, iPSC, and many more cell lines that are used for this instrument as well. In terms of the results that you get from the VIPS PRO, so the Acedin efficiency for a 96 well plate is above 70%. And as you can see in the figure on the left-hand side, this is maintained across 10 to 15 plates. And then also the clonal outgrowth is high because it's a low pressure based system. So we routinely see clonal outgrowth that's consistent over 10 plates as well. Once we have that single cell in the droplet in the bottom of the well and we've added media to the plates, the VIPS PRO would then perform the whole well imaging. And that's the image that you can see on the left-hand side. It's a whole well image of a 96 well plate. And you can see within that well, there was a single cell detected, again by artificial intelligence, that's labeled that well as clonal. And if there were 2 cells detected at that point, it would also highlight that as having 2 objects within the well. And again, the neural network used for single cell detection in the whole well is trained on a variety of different cell types, those being CHO, HEC, iPSC, and more. And there's also high accuracy cell detection. And this really supports the user when they're going through their images and analyzing the images. It speeds them up and makes them do that quicker. Once we've highlighted the wells that are clonal, we then have different AI-based neural networks that then can track the confluence of the wells at any given point in time. So the example that you can see on screen now is looking at day 7 to day 14. As you can see, the confluence or the colony growing out. And the colony, of course, is not green, but that's the overlay that this software has applied to that well to show where the colony is filling the plate bottom. And again, this is accurate. It doesn't pick up the well wall. And you can see against a human operator that's validating the accuracy of that neural network, it's about 95 to 99% accurate throughout different confluences. And again, this is also applicable to different cell types, such as the HEC and iPSCs that you can see on the right-hand side there. So the real benefits of these technologies, particularly compared to manual techniques such as limiting dilution, is you get increased seeding efficiency, that being the number of single wells containing a single cell can be approximately 3 times greater than limiting dilution itself. And also, by onboarding the artificial intelligence, you're reducing the effort and time required to analyze those images, particularly of the whole wells for the presence of a single cell, meaning that you need less plates and less FTE per cell line development project. Okay, so I'm just going to hand over to Sakti from CCRM now, who's going to provide a case study of how they've used the VIPS Pro in practice in human PSCs through to epithelial cell lines.
Thanks, Ryan, for the introduction. Hello, everyone. I'm Sakti Murthy. I'm a lead scientist at CCRM. I would like to start by thanking Adam for the great overview of the Solentim technology. It's a pleasure to be part of the joint webinar with Nova Biomedical, where we are showcasing some of the CCRM's recent work in clonal cell line development. Today, I will share our experience in generating clonal lines from epithelial lines and how this compares to our more established iPSC cell line development program. So I can give a quick introduction about CCRM. CCRM is a not-for-profit organization. It is established in 2011, and we are located in Toronto, and we are at the core of the Discovery District, providing a strong ecosystem to accelerate cell and gene therapy-based therapeutic development, supporting projects from early concepts through commercializations for both academia and industry. Our Toronto facility has the PDL lab, QC, GMP suites located on the single site, enabling a strong cross-functional collaboration overall for the project success. And in 2024, Omnia Bio was established as one of the first large-scale cell and gene therapy manufacturing facility in Canada. So it features like 26 clean rooms. CCRM, being the parent company of Omnia Bio, is supported through early clinical development. And once the program matures, most of the clinical and manufacturing related works happens at the Toronto Hamilton site. And this month marks the 50th anniversary of CCRM establishment. And during this 15 years of journey, CCRM has established creation of new therapeutic companies and also advanced the development of innovative therapies. So within CCRM, we have three prominent platforms: immunotherapy, stem cell therapy, and viral and non-viral delivery systems. The stem cell therapy platform is further supported by three core groups: reprogramming, gene editing, and differentiation and scale-up. And we also offer commercially available GMP IPP lines under the Linea Bio banner. And for the immunotherapy platform, supports the development of immune-based therapeutics such as CAR-T therapy, allogenic and autologous therapies. And the viral non-viral delivery platform supports the development of genetic modification tools. Together, these platforms serve as the three pillars supporting the CCRM's mission and vision. So this slide shows the overall workflow of gene-edited cell line generation. The process starts with transfection, which can be a viral or non-viral mediated gene delivery system, including like a CRISPR-based methods, transposons, and other delivery approaches. Based on the delivery method, the cells are given a sufficient time to recover before moving into the single-cell seeding using the VIPS platform. So once the clones mature, these plates are further screened using our high throughput screening processes based on the project requirement, which has a capability to screen thousands of clones in a quick turnaround time. So once we select the desired edited clone, this clone is further scaled up using our scale-up process based on the cell type, either using a cell stack or a bioreactor to generate millions of cells. And these are further being cell banked and further will be prolonged to a QC testing. So compared to our RUO and enabling airflow, like most of these GMP required reagents are kind of from initial time onwards, we kind of make sure that are all under a GMP compliant reagents to ensure that the process is easily transferable from early development to a clinical manufacturing facility. However, in the IND enabling workflow, we do have a rigorous documentation right from the initial stage development, where we make sure the MSAT makes sure the reagents and the processes are kind of having a high control and ensuring the quality of the product is high throughout the process. Also, we also have end-to-end quality oversight in the overall workflow, making sure the product is fit for the GMP grade manufacturing. So we have been using Solentim single-cell cloning technology for the past 5 years to generate clonal lines supporting both RUO and IND enabling programs. So we have successfully integrated this platform using multiple upstream processes, including viral and non-viral mediated gene editing. And we are also used for our reprogramming program as well. So we, across different multiple applications, what we have seen is we have consistently achieved a high single cell seeding efficiency as shown over here. And our overall cloning efficiency across these different programs, we are seeing around like 40 to 60%. And we have successfully incorporated the VIPS workflow into multiple of these programs, and we have reduced the timelines of these projects by at least 4 to 6 weeks, primarily eliminating the need for additional analytical testing to confirm clonality. And obviously the VIPS Pro also has 21 CMR compliant, which also serves as a good documentation, traceability documentations for the GMP expectations. So after the successful generation of clonal iPSC lines, our focus expanded into other difficult to clone types of epithelial origin that are significant interest in cell and gene therapy. So epithelial cell lines such as HEK and REP lines have been widely used for example, viral vector production, stable cell line generation, and regenerative medicine application. Therefore, establishing a monoclonal line forms from these systems can greatly improve the robustness and scalability of the process while reducing the batch-to-batch variability. This in turn enhances the long-term consistency and stability of the final product. And from a regulatory perspective, monoclonal cell lines are highly preferred over the polyclonal ones as they provide improved traceability and characterization of the product cell lines. So in the focus of the talk, I'll walk through our recent work on clonal epithelial lines, focusing on 4 main areas. First, on how media optimization really drives the successful outgrowth. And second, I will follow up with some of the data from the cloning efficiency, how it looks like across different cell types. And third, some of the challenges that we face with cell detection when working with some difficult cell types in single cell cloning. And also we'll provide some of the solutions to overcome these hurdles. And finally, we also show some examples and how we have expanded to use VIPS beyond the clonality of assurance. So as I mentioned to you, the media optimization for a single cell seeding is a very important step for a successful generation of monoclonal cell lines. I would like to draw your attention to this panel in the bottom for the fibroblast. As you can see, the media conditions in the M1 and M2, when we kind of compare these two, like where we kind of, the tech transfer had the media condition 1 and we optimized for the media condition 2. And we can see as such increase in growth rate by 2.5-fold. This is what we are generally look for a single cell seeding system where we opt for a high growth efficiency when the cells are single cell seeded. But often times, like not all the media conditions will have the striking differences in the early stages when we test them. And for example, like for these cell types like iPSC and HEK and REP, we went on do the single cell seeding to understand which is the best culture media to grow these cells in a single cell conditions. So as you can see from the iPSC and the HEK lines, the C4 for the iPSC and the S1 for the HEK shows a better growth rate compared to the other conditions. However, for the RPE, compared to all the conditions that we have tested, none of them stood as a very strikingly different from the other. So in these cases, what we generally do is like we check which our condition has given us a good growth rate, and we are able to get like sufficient number of clones from these testing conditions. We took that as the best condition to move forward for the single cell seeding. So from this slide, you can see that like the ones which are in the green highlighted ones are the media conditions we kind of took forward for the single cell cloning. And this slide shows the seeding efficiency and cloning efficiency across different cell types that we have tested and developed so far. And you can see for the seeding efficiency where it's able to print a single cell into 96-well plate. All the 3 cell types had a same seeding efficiency, around 70 to 80%. However, the cloning efficiency, that is from the single cell seeder, how many of these wells were able to have a colony, it kind of varied based on the media conditions. And iPSCs, you can see, is the one very well optimized, and we can see a 60% high cloning efficiency. And in terms of the HEK cell types, we saw like a 40% cloning efficiency, and for RPE, we saw a 10% cloning efficiency. And compared to the immortalized and stem cells, RPE cells are showing a limited expansion in 96-well plates, which may be a very inherent biological characteristics. So in this case, even with these 10% cloning efficiency, we were still able to generate clones and we were able to cell bank. The only caveat of these having a low cloning efficiency, this would need a lot of plates to be seeded and screened to generate the desired number of clones. So next we move on to understand how accurately the Solentim technology were able to detect cells across the 3 different cell types that we are demonstrating here. And as you can see from the bar diagram, the seeding efficiency from these cell types like were around 70 to 80%. And we can see these cell types were also having a diameter of around 14 to 17 micron size. And we are seeing that the precision in the single cell detection among these 3 cell types were also 98%. And from our experience, we have noticed that whenever we see a cell type which is having a diameter around this particular range, we know what range it generally works well for the single cell seeding, thereby eliminating the need for testing different seeding density as such. So overall, for the seeding efficiency, we didn't see much of a difference in the single cell detection. The accuracy seems to be 98% for all the 3 types. However, when these cells were growing into a colony, we saw some differences between the cell types. For example, these one are the whole well image, and the ones are the contrast image of the magnified colonies that are being expanding. And you can see for the iPSC monoclonal, these are very compact colony. And you can see the masking is quite 100% on the clones, having a very accurate detection of the colony confluency. However, for epithelial monoclonals, these are quite migratory in nature. And you can see the edges have like the single cells growing as a like monolayer. And you can see the masking in the middle where the colony is being like tightly packed, having to have an accurate detection. However, these single cells over here are not being detected well. So these images have been scanned using precision confluency, and Adam will be showing after the end of my talk, like what are the recent developments NOAA has done to improve the cell detection for these difficult cell types. So as I mentioned to you, epithelial cells are quite migratory in nature. So sometimes it poses some of the difficult challenges to prove clonality. For example, here, this is the day 0 single cell seeding timeline looks like, and this is the day 12 colony outgrowth looks like. And I have circled the wells where it's been single cell seeded, but on the day 12, you can see more than 2 colonies appearing in the wells. So if you just look into roughly the images, you may wonder that, oh, this is something like a cross-contamination coming from somewhere. However, when we do like periodical scanning of images, we do see these cells quite migratory in nature. For example, here, the day 0, you can see the single cell seeding over here, and that's growing into a nice colony until day 7. And when you do the day 10 scan, we saw a small one single cell being migrating over this inside the well and then growing into a separate colony. So at the end of the scanning system, you would see like 2 individual colonies coming up. So if we kind of miss this window, then we are quite kind of compromising the clonality assurance of these colonies. So that's why what we do generally is we introduce a pre and post media change imaging to improve traceability that strengthens the data confidence on this clonality scans. So another thing that we kind of generally do for establishing monoclonality, especially for cell lines where we are aiming for complex edit, where it's a kind of a rare to find clones. We also kind of use some of these multi-seeded cells to ensure that we are not missing out any valuable clones from this mixed populations. So how you generally do is using a time-resolved clonality imaging, that is having images in the early time point of scans, will ensure that if there is any single cell cloning coming from that particular mixed well seedings, that could be still used as a means of like using those traceable clonality documentation, we still be able to use those clones. For example, over here, this is an iPSC single cell seeded well. Like you can see, there are 2 cells being seeded, so it's an overseeded well. And when we do the day 0 2-hour scanning, that's the early time point scan, we can see only a single cell is being active. And we can see from the other subsequent scans, this particular cell is growing into a colony. So the other cell may have been just removed from the expansion process when we do the media changes. So for iPSCs, we do a daily media feed. So in that way, any floating cells would be removed from the wells. So using those early time point scans, we are able to prove even with the multi-seeded well, like they are coming from a single cell clone. So this assures that clonality assurance for these type of multi-seeded wells. So this gives us like a recover for high value clones from the overseeded wells. However, when we try to establish the same approach for epithelial cell lines, when we look into the multi-cell seeder to understand whether we could recover these clones if they are very valuable, we see here for an example that we are showing over here, this particular well has 3 cells. And we can see on the early time point scans, all the 3 cells are still attached to the well. However, only 1 cell is actively dividing. The others are still lingering. We can see they are not dividing. They have reached senescence. And we even see when the time progresses, only one of the single cell is kind of growing into a colony, and the others are kind of reached senescent, and then but they are still kind of lingering within the well. You can see even on the maturity day, only that particular into a colony, the single cell is still lingering into the well. So this kind of gives in a regulatory aspect a pretty drawback, because we can't prove monoclonality through the scanning alone. So we would require additional analytical testing to prove the monoclonality in such cases. So I just want to focus on the 4th focus area of my talk today. So apart from using VIPS for the clonality assurance, we also found some of the applications for using this particular instrument, especially for programs where we can't use any additional assays to understand the confluency. We kind of use the VIPS Pro for providing a clonality for these growing colonies. Compared to the automated microscopy, we find this gives like a whole well image view, where automated microscopy would always generally give a multi-field images where we need to stitch them to understand what is a whole well view looks like. And for the VIPS PRO, within 6 to 12 minutes, we can have like a whole plate imaging from a single plate. However, for the automated microscopy, they are always on the lower speed. When you think about like resolution wise, though they are moderate, when you kind of compromise for the resolution and we kind of prioritize for the high throughput whole well imaging, VIPS PRO looks to be very advantageous, especially for some of these applications, for example, NGS-based assays, where you want to understand what is the confluency looks like before sending for sequencing, or for even deciding the cell-based assay to make sure that like all the wells are having the same confluency before starting an assay, and even from functional genomics and optimization workflows, where we want to look into the confluency before understanding whether they are in the right confluency before initiating an assay. So I'd like to give some examples, like how we have used with application beyond the clonality assurance. So the figure over here is the cells that have been seeded for an NGS-based assay, and we would always do a clonality check, like confluency check, to make sure that all these wells are having the same confluency before sending them for sequencing. And we also have tested the detection of these wells based on different seeding density to see like how well the cells are being detected using VIPS PRO, and we are able to see differences between different cell densities, assuring that cells are being kind of consistently being detected using a VIPS PRO. So this is a very high throughput way we are able to use it to understand whether they can be used for the downstream process or not. So with this, I would like to summarize the presentation. So CCRM has optimized a robust single-cell cloning platform for iPSCs and has the capability to establish a single-cell cloning solution for other therapeutically valuable cell types. We observed the cell detection, accuracy of the single-cell seeding 98%, and through the precision confluency, we can detect robustly compact colonies. We also demonstrated how crucial media optimization is important in establishing a robust scalable cell line development, and also showed how to utilize the scanning features to improve the confidence on the clonality of the monoclones. That would support the regulatory alignment for IND enabling workflows, providing required traceability documents. With this, I would like to hand over to Adam to talk about some of the recent developments Nova Biomedical has done to improve cell detection.
Perfect. Thank you, Sakti. And yeah, thank you ever so much for giving that really insightful overview of how you've used the VIPS PRO in your workflows for iPSCs through to epithelial cell lines. As Sakti mentioned, what we're going to move on to next is how we've developed new technologies and new artificial intelligence algorithms to image and visualize those cell lines that are harder to see in the context of cell line development. So just to give you an overview and sort of adding to what Sakti mentioned within her talk, the standard confluence methods can often struggle across the board with low contrast and complex cell morphologies. And by complex cell morphologies, I mean if they're not circular and they're elongated or they're spindle-shaped nature. And with low contrast, it means there's no contrast between the background gray and the cell itself. This can lead to poor visualization and also the accuracy of the confluence quantitation can be lower with artificial intelligence models trained on other cell types such as CHO or HEC, which are vastly different morphologies to an epithelial cell line, for example. And because of this, we trained or created a new scan type, and this scan type is called the MSc Confluence Precision Scan. And this is where we used our confluence model, but this time we trained it on mesenchymal stem cells and MSc-like cells to deliver accurate confluence quantitation, particularly in those cell lines that are low contrast against the background and have that elongated spindle-shaped morphology. And on the right-hand side, or for all of these figures here, you can see on the left-hand side, the image in bright field of MSCs. The middle is how the standard confluence precision scan works that Sakti presented. And on the right-hand side is the improved detection accuracy with the newer scan type that's been released on the VIPS Pro. And if we look at a well that's been seeded with MSC-like cells, we have the same well on the image on the left-hand side and the right-hand side. And when a manual operator drew the overlay onto this well, it came out as 28.4% confluence. And you can see with the standard confluence precision, which detected at 10%, there was a difference of 17.5% here. But with the updated and newer available scans that are particularly trained on this type of cell type, the difference was much less, so only 1% difference between the ground truth versus the artificial intelligence. And of course, as I mentioned, this isn't just limited to MSCs, it's also MSC-like cells. And on the screen here, we have an example for a cultured meat application with this new scan type. And on the figures on the right-hand side, you can see what these cell lines look at a low density and versus a high density. And you can see the accurate overlay on both of those examples there as well. The next slide is just looking at the same scan type, but an ovarian cancer cell line with the MSc Confluence Precision Scan. And on the right-hand side, again, you can see this at low density seeding versus high density seeding and the accuracy between the two. And the final one is a mouse myoblast with the MSc Confluence Precision. And on the right-hand side, you can see how that performs for the high density seeding as well. The benefits of this is from your single cell through to the stable colony, the seeding, the assisted evidence of clonality, and then the MSc Confluence Precision Scans allow you to visualize the history of this clone through the high resolution whole well imaging and also the AI assisted analysis. Okay, so before we just go over for questions, I'm just going to do a brief summary of the webinar that we've presented today. So first up, the integrated single-cell seeding, imaging, and data management with the VIPS enable early high-confidence clonality confirmation. In the middle, we've got that scalable single-cell cloning workflows demonstrated stronger performance across cell types at CCRM, but epithelial cell lines require the optimized media and imaging strategies, as we've mentioned. And then lastly, the artificial intelligence enhanced imaging capabilities enable accurate detection and confluence quantitation of several different challenging cell types, including MSCs, but extending through to cultured meat, etc. as well. And this extends the applicability of the platform for improving confidence in clonality and growth assessment with some of the cell lines that Sakti's mentioned today. So thank you ever so much for coming along to the presentation today. On the screen, you'll see emails for both Sakti and I. If you do have any questions that you don't get answered during the Q&A or you want to reach out directly, feel free to shoot us an email and we'd be happy to get back to you.
Thank you both very much for the very insightful presentation today. At this point, I'd like to invite our audience to keep sending their questions or comments right now for the Q&A portion of the webinar. If you're attending via GoToWebinar, please use the questions window. And for those watching on LinkedIn Live events, feel free to post your questions in the comments section. Now, I've already received some questions from the audience, so we'll get ourselves started with those. And looking here, the first question I have, and maybe Adam, this was something you could share some thoughts on. They're asking, what enables earlier and more confident confirmation of clonality in this workflow compared to traditional methods?
Yeah, sure, I can take that one. So as I mentioned at the start of the presentation, when you're using the VIPS, you're putting an empty plate onto the instrument and you're generating a droplet with the VIPS of a cell suspension, and then you're imaging that droplet at 20 different heights to ensure that droplet contains a single cell. And this gives you high confidence at the point of seeding that well is a clonal well. And then, as Sakti mentioned towards the end of the talk as well, is you also have that time-resolved imaging, so from day 0 through to day 14 or up until the point of passage, where you can really track the outgrowth of that colony to ensure there's a single cell throughout that well at a given point in time. So you can increase the confidence on two fronts from that point of view.
Perfect, thank you. Now I've got another question wondering, where are the main efficiency gains in the workflow compared to limiting dilution?
Yep, so I can take that one as well. So compared to limiting dilution, where you're sort of statistically hoping there's a single cell within the well, the seeding efficiencies for the VIPS Pro, so the amount of wells containing a single cell, depending on the cell line, will be somewhere between 3 to 4 times higher than limiting dilution. And then in addition to that, you also have the artificial intelligence with the cell detection networks that's reducing the amount of time taken to review those images. So you have more cells in less wells, and it also takes less time to review those wells.
All right, perfect. Thank you. I've got a question that's come in. They're asking, I have a question for each of you. To Sakti, they're wondering, how is cell banking organized? How do you ensure the traceability of the manipulations, and is there any particular software used?
So we do have a centralized QMS system, which is like a quality management system that maintains the overall traceability, and we have a quality assurance. So generally we have like a master batch records created for each of these processes from beginning to the end. And for cell banking, like that's a kind of like a very standardized program we have for iPSCs and different cell types, that we call it as like a very mature program. In our organization, like we have done iPSC, mesenchymal stem cells, and CAR-T cell banking. And we do have like a fill and closed system for fill and finish. So we kind of, for each, depending on the cell types, we have bioreactors that can handle like generating millions of cells based on the cell types. And we have like a regular traceability documents right from the beginning to the end, and making sure these master cell banks that has been created in this process meets the required criteria. And even before beginning itself, like we also ensure that all the in-process testing has been fully QC validated, making sure at the end of the process we have a fully validated cell bank that's been ready for a QC release testing.
Very good. And Adam, the question I have for you is, which features make VIPS Pro GMP compatible technique?
Yeah, really good question. So there's 2 elements of this. Firstly, first up for the consumables. So in terms of the consumables for the VIPS Pro, it's really the single-use seeding kits, so the cell reservoirs, that are the things that come in contact with the cell culture itself. And these come in 2 formats. Firstly, a research use only format, but secondly, a GMP compatible format. And the difference between the 2 are really the testing and documentation that's supplied with each different type of kit. And I'd be happy to share this with the individual that's answered the question as well, just giving an overview of what documentation we provide for the GMP compatibility. And then on the other side of it is the software and the security behind the software. So as we mentioned, the STUDIUS software is 21 CFR compliant, and this means it's compliant in a range of different manners. Firstly, from having to require a password to get onto the system, how the data is stored and encrypted, and also there's optional features where you can have audit trails produced by STUDIUS to tell you who's interacted with the data and edited the data, and give you an overview of how that data has been handled throughout the cloning program and beyond.
Perfect. Wonderful. Thank you very much for that answer. Next up, Sakti, I've got a question for you. Someone is wondering, why is establishing monoclonality more challenging in epithelial cells compared to iPSCs?
So The way the epithelial cells grew within the cells they are quite migratory in nature compared to iPSCs. iPSCs tends to form like a closer compact colonies, but epithelial tends to grow like a monolayer, and they have like a quite migratory in nature. So sometimes it's challenging to establish monoclonality if we kind of miss the scanning window. And we often see that during the media change conditions, we see that like some of the cells lift off from the colonies and migrate within the well, and then they place into another region of the well and then start to grow into a separate colony. So if you miss that where that single cell is coming, if you don't have that scanning data coming from where that single cell within the well is coming, then it's very hard to prove, to understand a high confidence on that clones. So this becomes very challenging in terms of iPSC versus epithelial cells. So, and also iPSCs, we do routinely media changes, and none of these cells stick to the well after media change. However, epithelial cells have a very sticky nature. Even like once you can see that the cells migrate and then stick to the inside of the well, making it hard to understand where the cells are coming from. So that's what we need, that periodic scanning to ensure that these are coming from the clonality documents are in place for the IND enabling purpose.
All right, perfect. Thank you very much. Next, then, I've got a question for you, wondering how does cell type affect clonal outgrowth, and what role does media optimization play?
So media optimization is the foremost important thing, and that's the first thing we try to develop to ensure that we have sufficient clones at each end of the single cell seeding process. And compared to iPSCs, epithelial cell lines are just kind of in the early stages of the development, especially for the retinal pigment cells that we are currently working on to progress the cell line development for optimizing the media. So if we don't have a optimized media, then we are already kind of starting with a little bit of drawback in terms of then we need to have more plates need to be seeded so that we have sufficient clones required for the downstream processes. So remember, for gene editing, at each and every stage, we do analytical testing, making sure it is clean edit and it doesn't have any off-target site editing. So having that number of clones at the end of the single cell splitting will give us some advantage, so that even if you lose some of these clones, based on the analytical testing, we'll be having some for the cell banking stage. So that's why like media optimization is crucial for the success of the single cell ciliating.
Perfect. Excellent. Thank you very much for that answer here. Another question that's come in is wondering, what are the practical advantages of whole well imaging in VIPS compared to traditional automated microscopy?
So it always depends on what is the application that we are trying to use. So we kind of did like an accidental discovery of using an extended application of VIPS. Apart from clonality assurance, what we could do. So we are looking for options where we can do like a whole well imaging, especially for bright field images. We know that for cell images that has been fluorescent protein or stained, like it's very easy to image using a traditional microscopy, and then it also requires a lot of field images for a traditional microscopy to pick up the whole well image, compared to VIPS Pro, which can do with a single shot, like a whole well imaging can be done with this precision confluence scans. So this is kind of like an accidental discovery, and we kind of used it for a lot of applications, including NGS-based methods. And some of these assays where you would be able to not use any viable dye, we also use a confluency-based scans to understand the cells confluency to be using as like a parameter to understand the production of these clones.
Perfect. Thank you very much for that answer, and both of you for all of your answers today. But we have reached the end of our time now. If we couldn't attend to your questions, know that the team at Nova Biomedical will follow up with you. Or if you have some further questions, you can direct them to the email addresses that are up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen when you click exit, and your participation is appreciated, as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com so that you can register for this event and then gain access to features such as viewing that recording and more. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you, everyone.